In this video module, you will learn about microscopy and the various staining techniques that we use for seeing bacteria. The size of bacteria are measured in microns or micrometer. It is denoted by mu symbol. Now, suppose you take a ruler, a standard ruler, which is marked in centimeters. The first centimeter is divided into 10 divisions. You have 10 lines. Take the first two divisions, divide it by 1000 and take the first division in that. So that is 1/1000th of a millimeter. The size denotes a microns, which is used to measure bacteria. That is 1/1000th of a millimeter. Now again divide this micron into 1000 divisions. Divide it 1000 times and take the first division. That is 1/1000th of a micron, which is the size called as a nanometer. So a nanometer measures 1/1000th of a micron. Nano means anything small, just like the nano cars that we see on the roads. Now this is the size that we use to measure even tinier organisms which are tiny than bacteria, like the viruses. Now let's say you divide this nanometer into 10 divisions only and take the first division, that is 1/10th of a nanometer, which is called as 1 Armstrong unit. So you can say that since bacteria measure so small, we need to give them a good magnification to be visible, to be made visible to the naked eye. For this purpose, we use microscopes. What you see here is a light microscope, a simple one. It's just an optical microscope. There are many types of microscope. The basic principle is magnification. Here in this picture, where you see a light microscope, the part marked 1 is the eyepiece, wherein you put your eye to see it, and then 2 is the handle, and 3, there are actually 3 lenses, 3 lens systems. They are called as the objective lens system. Now 1 and 3, that is the eyepiece and the objective lenses, magnify any object which is smeared on a slide and put on the stage. So again, below the stage, you can see marked 4 is the condenser lens. So this condenses all the light and makes it to pass as a single beam of light below the stage. So actually the magnification is a product of the magnification of the eyepiece and the objective lens system. To see bacteria, we need to stain them to make it easier to be seen through the microscope. There are many staining techniques to be made visible with a light microscope. To see objects which are microns in size, we use the most common and the simplest technique, that is the Gram staining technique devised by Hans Christian Gram. As you can see, there are essentially 4 steps. Step 1, 2, 3, 4. Step 1 is the addition of a dye, and step 4, the last step, is also an addition of a dye, a stain we call in microbiological terms. The first dye is blue, violet in color. The last dye is pink in color. The violet color dye is the methyl violet. This stains all the organisms which are put on a slide from a smear on the slide as violet in color. There are many types of organisms, so when you use it once, it stains all the organisms as violet. The next reagent is Gram's iodine. When you use it, it helps to fix the violet colored dye, that is methyl violet, onto the cell wall of bacteria. Now, if you were to peep into the microscope at this stage, all the organisms, all the bacteria will appear violet in color because the cell wall has been fixed with this dye with the help of iodine. It is a fixative agent. We call it as a mordanting agent. The next step is like an eraser. So that is, we are using alcohol or acetone. That depends from lab to lab, it varies. Now this is an eraser like step, a rubber like step, wherein when you use this decolorizing agent, some organisms let go of the stain and get decolorized or become colorless. This step is crucial. It helps to differentiate between those organisms which are able to retain the violet color and appear violet, and those organisms that get decolorized and lose the color. The next step is addition of the second dye, that is the pink colored dye, as a counter stain. That is, here we use a dilute solution of carbol fusion. So here those organisms that have lost the color take up the second dye and appear pink in color, whereas those organisms which have held on to the color and not got decolorized retain the original violet color and remain violet. So the violet colored organisms which have managed to retain the color, they are called as gram positive. Those organisms which have got decolorized and taken up the second dye is called as gram negative. So here the third step, that is the decolorizing step, is very important to decide whether an organism can be gram positive or gram negative. So the pink color dye, the organisms here we see are gram negative, and the violet color organisms are gram positive. Here the gram positive organisms are spherical in shape and the pink color organisms are rod shaped. It can be either way. So you see a picture of a gram stain smear in the next slide, and observe it and comment on it. I'll give you 30 seconds to observe. What you see here, when you see a gram stain slide, you have to remember to comment on it under 3 headings: the shape, color, and the arrangement. Have you seen 2 types of organisms? Yes. One is violet in color, another is pink in color. So now you can say what are the violet color organisms? Gram positive are violet in color. So under shape you can write, under color you can write as violet. The other organisms are pink in color. So under color you can write it as violet, pink in color. Now regarding the shape, you can see the violet color organisms are spherical, so they can be called as cocci. The pink color organisms are rod shaped, so they can be called as bacilli. Regarding the arrangement, the violet color organisms are arranged in groups or clusters, whereas the pink color organisms are scattered in arrangement. So we have observed all these shape, color and arrangement. Then we infer that the type 1 organisms are gram positive cocci in clusters. The type 2 organisms are gram negative bacilli in a scattered arrangement. This is the simplest staining technique. Another differential staining technique like the gram stain is the acid-fast staining technique. The popular method is Ziehl-Neelsen's technique, named after the persons who developed it. So this is used to stain organisms like the Mycobacterium tuberculosis. What is meant by acid fastness? It's like the organisms hold on to the primary color without losing it, even if treated with acid. They are acid fast. So here on the left hand side you can see there are it's essentially a 3-step technique. Sometimes it can be modified into 2 steps also. Here again we use the same principle: a primary dye, a decolorizing agent and a secondary dye or a counter stain. The primary dye here is a strong solution of carbol fusion. You can heat it, so all the organisms will take up the primary dye and appear red in color or pink in color. Again the next step where you use a decolorizing agent. Here it's a strong solution of acid. It's a strong acid. Even after using acid, it doesn't get decolorized, retains the color and appears red. Those organisms who have lost the color or got decolorized take up the second dye and appear blue in the next step. So those organisms which manage to retain the original color and appear red are called as acid fast, and those that haven't got which are not able to retain the color are non-acid fast. So in the next slide you see the acid fast staining technique, wherein you see a blue background against which the acid fast organisms appear pink in color. It takes a deep pink color marked by the arrows. Thank you.